Alright, so here we are on lecture 10, and I'm going to go through the plasma membrane structure in this one. So remember that plasma membrane is going to bounce backward here to this figure. So plasma membrane is referring to this outer membrane that basically defines the outer boundary of the cell. Okay, so here this purple thing that surrounds, and of course this would be an animal cell. A plant cell would have its cell wall that would be on the outside of this, but the plasma membrane would be this cellular membrane here. Okay, so as it defines the outer boundaries of a cell, it is obviously also going to be the interface through which the cell is going to interact with its neighbors, whether that's through contact or whether the cell releases something that then interacts with the surface of the other cell. Basically everything that happens between the cell and the things that are going on around it are going to go on across that plasma membrane. Now, there will be some things, as we'll see, that can actually get across the membrane and inside and interact inside, but most things are going to interact with the cell at this surface. So let's think a little bit about what we know about the membrane structure. You guys will probably remember from introductory courses where we talked about phospholipid bilayers. So obviously the most important and abundant component in a membrane is what's called a phospholipid. This is a very specific structure, and I'll show you a picture here in a second. One of the most important pieces or properties of the phospholipids is the fact that they are amphipathic, and you'll recognize that amphi meaning both, and pathic means that you are, remember, sympathy or empathy, it refers to feelings, but in this case, of course, it's referring to whether you interact with an aqueous environment or not. Do you interact with it, or do you not? And so a molecule that is amphipathic, like phospholipid, is going to have an area that is hydrophilic and an area that is hydrophobic. So the hydrophilic portion is what we call the head portion of the phosphate, and has a phosphate group and some other group attached to that. And the backbone is going to be this little 3-carbon structure. So one of those carbons will be attached to here, or this phosphate group with its hydrophilic head, and then the fatty acids, these long hydrocarbon, which are very hydrophobic, are going to basically be attached to the other 2 carbons, and those create what's called the tail. Different fatty acids on the phospholipids can obviously alter the properties of that membrane. So the fatty acids can vary by the length, the number of carbons they have in them. You can introduce double bonds in one that actually controls the nature of that fatty acid as well. And I already mentioned how the smoothie R, which is part of the endomembrane system, is largely in control of what's going on with membranes. Other components of the membrane will be contributed by other organelles as well, and largely the nature of the plasma membrane is going to be responsive to things that are going on outside, and to a certain extent the temperature that the cell finds itself in. So the plasma membrane is not just a structure that stays the same all the time. It's very dynamic, and the cell will change it depending on the conditions it finds itself in. Alright, so here is a phospholipid, and here you can see, well, you can kind of see there's 3 carbons here, and this would be what we call it's called glycerol, this thing right here. Notice there's 3 carbons. Then you got the phosphate attached, and then there's going to be some kind of a hydrophilic group here. So the kinds of hydrophilic groups we have would be these choline and serine and ethanolamine, and there are several others. These would be what have the hydrophilic groups in place. Notice they have charges, and so those very, very nicely interact with an aqueous environment. And then the other 2 carbons are going to be attached to what we call the fatty acids. And notice CH, 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 all the way down those hydrocarbons. And the length matters, and then something like a double bond will actually introduce bends to these fatty acids. And if you put 2 double bonds in, of course it'll it'll kick twice, almost like a dog leg. The nature of phospholipids, and remember this will just happen spontaneously if you put them into the aqueous environment, they are actually going to assume what's called a bilayer. And one of the reasons why is that the phospholipids, you remember I showed you a micelle several lectures ago, and in the context of the micelle, those fatty acids that are going to create a micelle tend to be fairly shorter and not have these double bonds in them that create the bends. Now phospholipids, the fatty acids are much longer, so that if you were to actually draw a micelle, or draw a micelle on fatty acids, they just don't lay together very well. There'd be too much space between them as they kind of almost like slices on a pie. Because of the tails are so long, it pushes those hydrophilic heads so far apart that they just are not stable. So instead, they form what are called bilayers, where you're going to end up with a surface that is hydrophilic and another surface that's hydrophilic, and then the zone in between where the two tails interact with each other, that is going to be hydrophobic. And of course, if you create a bilayer, then the only way for that hydrophobic layer to completely be stable is if you enclose it all the way around and actually create a spherical shape. So here's a figure out of a pretty old cell book that I used to have in college. I just like this figure because it really shows that. So here would be what a bilayer might look like. So you've got the two hydrophilic layers, and both of them have the hydrophobic tails facing each other. And the only way that you are not going to have an edge exposed is by folding the whole thing down and around so that it forms a sphere, and more importantly, it's a hollow sphere where there's a compartment inside, and of course you'll have the outside layer as well, or environment as well. Each one of these layers is what's called a leaflet, and we won't really worry too much about them, except just remember that each leaflet is independent of each other, and those, as they are managed, you're going to have one leaflet that faces outside, you're going to have one leaflet that faces inside, and the cell can actually change the components of those leaflets to match which direction they're facing. Okay, so here's just a kind of a drawing of it, where you've got the outer leaflet, the inner leaflet, and this would be one aqueous environment, and of course the outer aqueous environment. And this is important because this would be the primary way in which a cell now has an area where it can control what's going on. And so it can now transport things in that it can hold on to and no other cell can have, or it can also transport waste out and protect itself on the inside. The contents of the plasma membrane is what we're going to call the cytoplasm. Of course, cytoplasm includes everything that is inside the cell other than the nucleus, so the organelles and what we'll call the cytosol. So we'll talk about that in a little bit in a later lecture. And then everything outside would be these two environments. All of the internal membranes create those internal compartments we know as the organelles. And as far as the barrier goes, this thing is really effective against large molecules. This is very effective against polar molecules, or charged molecules are not able to get through that hydrophobic zone either. Now, anything that's small can generally get through. Anything that is hydrophobic can generally get through. And so if you think about it, what we have here would be I think this was a physiology book picture, and you can see here's the bilayer, and the bilayer would be very permeable to things like gases, so CO2 and O2 just are going to move across really easily. Small molecules like alcohols will move across very easily because they're not charged. Water, actually moves across fairly well as well, although not quite as quickly as the smaller gases. So even though it's polar, it's small enough to slip through. And if we need to move large amounts of water, then we have to use other mechanisms. Glucose is actually small enough to kind of get through, but we really do need transport mechanisms to get the amount of glucose we need for making ATP. And then, like I said, things like charged molecules or charged atoms, and large things like amino acids and nucleic acids and polysaccharides and things like that. These guys are not going to make it across. So peptide hormones, growth factors, all of these kinds of things are going to have to interact with the surface. So when we think about cell, and remember we're talking about all of those interactions that are going to go on around it. If we are small and we are nonpolar, we're going to be able to go on inside and interact with the cell inside. But if we are stuck outside, then everything we do has to go through this structure of the plasma membrane. And so that's why this plasma membrane is such an important piece to development, because this plasma membrane is the surface by which we are going to interact with everything around us. And those interactions are going to determine what we do developmentally. Okay? All right, so we're going to talk a little bit more about the structure of this guy and how that structure is going to then open us up for being able to discuss some of the functions in the lecture after that. All right, so talk to you later.